In this lecture, we are going to learn about the antibiotics that inhibit bacterial cell wall synthesis. Now, majority of the bacteria are surrounded by a thick cell wall, and the cell wall is composed of peptidoglycan. So, the cell wall is composed of peptidoglycan that provides stability and rigidity to the cell structure. It also helps to maintain the shape of the bacteria and does not allow the bacteria to swell and prevents death due to lysis. Now let's see the composition of bacterial cell wall. The bacterial cell wall is composed of peptidoglycan, which consists of parallel strands of glycan, parallel strands of polysaccharides, which consist of amino sugars, namely N-acetylglucosamine and N-acetylmuramic acid. So N-acetylglucosamine and N-acetylmuramic acid is linked to a pentapeptide chain consisting of amino acids. And these peptide chains are further cross-linked with other peptide chains by a pentaglycan bridge, and this is known as cross-bridging or transpeptidation reaction. And this transpeptidation reaction or cross-bridging provides strength to the bacterial cell wall. So the process of cross-bridging is catalyzed by enzymes known as penicillin binding proteins. So the penicillin binding proteins are expressed in the cytoplasmic membrane of the bacteria and catalyzes transpeptidation reaction or cross-bridging that provides strength to the bacterial cell wall. So now that we know the composition of peptidoglycan, let's see what are the different steps in the synthesis of peptidoglycan and the various antibiotics acting at different steps of peptidoglycan synthesis. So we will learn the site of action of various antibiotics that inhibit cell wall synthesis. So the first step is the formation of various amino sugars, namely N-acetylglucosamine and N-acetylmuramic acid. So N-acetylmuramic acid is formed in presence of an enzyme that is enol pyruvate transferase. So enol pyruvate transferase catalyzes the formation of N-acetylmuramic acid, and an antibiotic known as fosfomycin blocks or inhibits the enzyme and prevents the formation, decreases the formation of N-acetylmuramic acid, which is an essential component of the bacterial cell wall. So fosfomycin acts in the early steps of peptidoglycan synthesis. It decreases the formation of peptidoglycan monomers, that is the N-acetylglucosamine formation, and N-acetylmuramic acid formation is inhibited. Now the second step is inhibition of assembly of peptidoglycan monomers or inhibition of polymerization of peptidoglycan, which occurs in the periplasmic space. So how does it occur? The peptidoglycan monomers are synthesized in the cytosol, and a lipid carrier or a transporter known as bacterial is expressed in the cytoplasmic membrane. So it's a lipid carrier which is expressed in the cytoplasmic membrane, and it helps to transport the peptidoglycan monomers into the periplasmic space. So bacterial, which is a lipid carrier, undergoes phosphorylation into active form, that is pyrophosphate form, and it becomes active, and once it becomes active, it helps to transport the peptidoglycan monomers into the site where the polymerization of peptidoglycan occurs. Now, once the peptidoglycan monomers are transferred to the existing peptidoglycan chain, to the growing peptidoglycan chain, dephosphorylation of bacterial occurs, and this dephosphorylation is an essential step because this causes recycling of bacterial to its original site and is available for subsequent rounds of transport. So dephosphorylation is a crucial step. Bacterial loses its phosphate group and is once again available for the transport of peptidoglycan monomers into the periplasmic space. So there is an antibiotic known as bacitracin and cycloserine which inhibits dephosphorylation of bacterial. So since dephosphorylation is inhibited, bacterial is not available for subsequent rounds of transport. It is not available for the transport of peptidoglycan monomers to the site of polymerization. So the polymerization of peptidoglycan is affected, and in this way the cell wall synthesis weakens because of decreased peptidoglycan synthesis. The next step in the synthesis of peptidoglycan is transpeptidation reaction or cross-bridging. So prior to cross-bridging, there is cleavage release of the terminal amino acid, that is D-alanine. Now, glycopeptide antibiotics such as vancomycin and teicoplanin attaches, it strongly adheres to D-alanine and prevents the release or cleavage of D-alanine, and this inhibits cross-bridging or transpeptidation reaction. So, further transpeptidation reaction cannot occur because of the antibiotic being attached to the D-alanine and inhibiting the cleavage or release of D-alanine. Now, the final step in the synthesis of peptidoglycan is transpeptidation reaction or cross-bridging. As we have seen that the two amino sugars are linked to a pentapeptide bridge, and there is a cross bridge that is formed between L-lysine and D-alanine. So, this cross bridge formation occurs in presence of penicillin binding protein. So, penicillin binding proteins catalyze the transpeptidation reaction, catalyzes the formation of a cross bridge between L-lysine and D-alanine, and therefore antibiotics such as penicillin, cephalosporins, carbapenems and monobactams, so the beta-lactam antibiotics produce the action by inhibiting the cell wall synthesis, and the site of action is they inhibit the penicillin binding proteins. The enzymes are inhibited that catalyze transpeptidation reaction. So, because the cell wall is weakened, there is osmotic drive of fluid from the environment into the bacterial cell. There is swelling of the bacterial cell, and this causes lysis and death of bacteria. So, cell wall synthesis inhibitors act as bactericidal agents because they cause death of bacteria. They reduce the number of bacteria. So, a quick recap on cell wall synthesis inhibitors that act at various steps in the synthesis of peptidoglycan. So, phosphomycin acts in the early steps of peptidoglycan synthesis and inhibits the formation of amino sugar, that is N-acetylmuramic acid. Bacitracin and cycloserine inhibits the transport of peptidoglycan monomers into the periplasmic space where polymerization occurs. This is because bacitracin inhibits dephosphorylation of bactoprenol, which is a lipid carrier, and because dephosphorylation does not occur, bactoprenol is not available for transport of peptidoglycan monomers to the site of polymerization. The third group of antibiotics are glycopeptide antibiotics, that is vancomycin and teicoplanin, and these antibiotics act by attaching strongly to D-alanine and prevents the release of D-alanine, which is the terminal amino acid that is attached to the amino sugar. By inhibiting the release of D-alanine, the further transpeptidation reaction is inhibited, the cross-bridging is inhibited, and this leads to weakened cell wall and cell wall synthesis inhibition. The last group of antibiotics is the beta-lactam antibiotics, that is penicillin, cephalosporins, carbapenems, monobactams, that act by inhibiting the penicillin binding proteins. So, penicillin binding proteins catalyze transpeptidation reactions or cross-bridging. So, because of the inhibition of penicillin binding proteins, the cross-bridging or transpeptidation polymerization does not occur, and this leads to a weakened cell wall, death of bacteria, lysis of bacteria, accounting for its bactericidal activity. Now, lastly, let us see why penicillin is more effective against gram-positive bacteria as compared to gram-negative bacteria. So, this is because of the structural differences between gram-positive bacteria and gram-negative bacterial cell wall. This accounts or this influences the antibacterial activity. As we can see in this figure, in gram-positive bacteria, there is a thick peptidoglycan layer which is present in the outer side. So, because it is present in the outer side, the antibiotic can easily access peptidoglycan and can inhibit the synthesis of peptidoglycan. So, easy access of antibiotic to the cell wall, to the peptidoglycan, accounts for its greater antibacterial activity against gram-positive bacteria. Unlike gram-positive bacteria, gram-negative bacteria has a thin peptidoglycan layer that is sandwiched between outer membrane and inner membrane. The outer membrane consists of lipopolysaccharides and teichoic acid that act as barriers to the entry of antibiotics. So, the antibiotic is not able to penetrate. So, this decreases the permeability of the antibiotic to the peptidoglycan layer, so it cannot easily access the peptidoglycan layer. So this is the reason why penicillin is more effective against gram-positive bacteria than against gram-negative bacteria, because of easy access to the peptidoglycan layer compared to gram-negative bacteria where peptidoglycan layer is not easily available for the antibiotics. However, there are certain antibiotics such as amoxicillin, ampicillin that are hydrophobic in nature and they can penetrate the outer membrane because of the presence of porin channels. So there are porin channels present in the gram-negative bacteria that facilitates the entry of antibiotics such as ampicillin and amoxicillin, which can enter and inhibit the peptidoglycan layer that is sandwiched, that is present between the outer membrane and inner membrane. So in this video, you have learned about the cell wall synthesis inhibitors, the mechanism of action, the steps in the synthesis of peptidoglycan, and the site of action of various antibiotics. So thank you so much for watching. Please like, share and subscribe to this video if you find my video useful.